So hi, I'm Andrew Wilson. I'm a Senior Research Commercialization Manager at the University of Bristol. And over the past kind of 5 or 6 years, I've had the pleasure of working with some amazing scientists developing some amazing new therapies for patients. And I've really, I'm employed by the university to try and support those innovations from discovery at the university through to the point of partnering with a commercial organization from the progression. I'm sure you're going to hear this lots today, but the development and clinical testing of ATMPs is a very complex and expensive process, and not all of that expertise exists within our university. So we often partner with commercial organizations who have the resources and expertise to progress our therapies, and that's the role that I play at the university. James has already gone through some of this landscape, and my trial numbers don't match his. I got mine from the Alliance of Regenerative Medicine, so I'm not sure who's right. But it is a rapidly growing market. It's driven by the promise of even more transformative medicines hitting the market. The market size numbers for cell therapy are expected to reach 48 billion by 2027, and gene therapy 14.8 billion. And I think the important thing here is that they're all growing at 25% annually, which is significant. Large pharma continue to be acquisitive in terms of buying up smaller biotechs, and that in turn is driving significant amounts of VC investment. So I don't know if these figures are right, but there were $9.8 billion of VC investments in new companies last year. And universities play a really important part in this landscape. I'm not surprised to learn the majority of the trials that are ongoing at the moment are not company sponsored, so they're sponsored by the universities, healthcare organizations, or government. And also the majority of ATMPs in development were initially created at universities, so we have a very important role to play in the growth of the landscape. All of this means there are lots of opportunities for us to partner our early stage research with well-funded companies to rapidly translate it into new therapies. There's a lot of great research going on at Bristol. There's a lot of great ATMP research going on. Our first spin-out company was a company called Azalon, which was set up in 2008 to commercialize a cell bandage for meniscal tear repair. And then it kind of went quiet in terms of disclosures into my office. In the last 5-7 years or so, I've seen a dramatic increase in the number of new projects that we've got, cell and gene therapy assets, tissue engineered products, and enabling technologies. So we've had to really sort of work out how we can best support those projects. And they're across a range of indications and disease areas, including renal, CNS, ophthalmology, cardiac, and hematology. And that's led to the creation of a number of spinouts and also deals, notably Cytoseq. Carol's going to tell you a bit about them later. Mogrify, that started off as a software play initially, and PureSpring Therapeutics that we set up about a year and a bit ago in the renal field. Hopefully there are more coming soon. In fact, I know there are more coming soon. So what does my team do to support these? I think, I was talking to a couple of the other speakers earlier, and I think I've probably got the easiest job. What I do isn't actually that complicated. I just need to find great projects. And there are lots of those in the universities. People come and tell me about them all the time. And really, what I then need to do is just facilitate the progression of those projects. I don't do much myself. I link people up with the right people. I find money and push projects on the way. So once the project comes to us, we need to assess the commercial opportunity. And because of the commercial landscape, that's not terribly difficult in the ATMP space at the moment. We really need to focus on whether there's an unmet need and whether actually it's tractable. We then need to seek patent protection really for most ATMPs. Because they're going to be taken up by a commercial organization, we really need some patent protection in place so they can essentially protect their investment in running the clinical studies and then bringing the ATMPs to market. I think one of the most important things we do is kind of support early translational funding. We often get technologies that are very, very early stage, and they're a bit further away from where the companies want to see them. So we have to kind of work out what experiments do we need to do to take technology from where it is now to a point where we can partner it? And I don't often always know the answers to that. So what we'll do is we'll talk to people who do know the answers. So that's regulatory people, CROs, but we'll also interact early with companies and investors, really to understand what they need to see in a data package before they'll really start thinking about this as an opportunity. And all of that feeds into translational project planning. If we can answer all those questions the companies have, then there's a good chance that if we're successful with the project, then we can partner it. And assuming that all goes well, I then lead on concluding the deals from a university perspective. And really what we're focused on doing is making sure that our deals are structured so that the technologies can be progressed and create impact. And I'm just going to talk you through two projects that I've been involved in. One was a partnering deal and the other a spin-out. This was a very exciting technology. I think it may have been the first gene therapy project that I had to deal with. And a group at Bristol led by Andrew Dick and Dr. Colin Chu came up with a gene therapy for glaucoma. Glaucoma is a massive problem, causes 12% of all blindness. And Colin was very excited because he potentially thought he may have a cure for glaucoma for a single intravitreal injection that could be performed in an outpatient clinic. What I'd include a tiny bit of science. There's a graph, and it shows that when you inject the AAV, it reduces intraocular pressure, and that's a very clinically significant drop. How did I support it? The main thing we need to do is get data in a mouse model or 2 mice models, and we found some internal proof of concept funding to allow Colin to do that work. It was a single asset, which means it didn't really lend itself to a spin-out opportunity. So we talked to a number of clinical stage biotech, and I'm pleased to say we partnered it about 1.5 years ago to a clinical stage gene therapy company, and they're now progressing this therapy towards candidate selection. There were 1 or 2 problems, CRISPR being one of them, and that presented obviously a freedom to operate or significant IP uncertainty for the company. But nonetheless, we managed to do a deal on that. The next project I want to talk about is now a series of projects and led to the creation of a spin-out company called Pure Spring Therapeutics. Moin Salim, I think, was going to be here, hello Moin, and his group at Bristol Renal were the first group to show in vivo rescue of renal disease via gene therapy. And obviously this was really exciting. The team were really excited about it. But I didn't really know what to do next or in what order. So we reached out to the Cell and Gene Therapy Catapult Centre, and they provided some really useful support on the regulatory side of things, and also the non-clinical development planning and the order in which we need to do things, which was really helpful in helping us understand our thinking in terms of what next. There's quite a lot of commercial interest. Moin's very well connected in the renal community, and so we had people coming to talk to us and wanted to take the technology. And we didn't quite know what to do with it. All we wanted to know is we wanted to create as much benefit as quickly as possible. We had a number of conversations, but the conversation with Syncona was the most interesting. They had clear alignment on our vision for where we could take the platform, and that led to the creation of Pure Spring Therapeutics. That was set up about 1.5 years ago. They've recruited an amazing team, highly experienced, and now they're rapidly progressing a number of life-changing therapies for patients with renal disease towards clinic. Watch this space. So we've worked on a few projects, but we're always learning. But a few things that I think are important, is worth mentioning. So in vivo animal data is key if you want to partner. It's what commercial organizations really want to talk about, and it's what the investors really want to see. But apart from making the opportunity more commercially attractive, that data is really crucial to ensure that patent filings are robust. And robust patent filings really underpin the downstream translation and commercialization. It's really good to understand the FTO position around therapeutic constructs, but don't let it bother you too much. So I refer to the glaucoma example. That included CRISPR, and the capsid was actually not available for licensing. So we've got a proof of concept, but there's significant optimization work that the commercial partner now needs to do to circumnavigate those FTO issues. But that's really something a commercial organization is better off placed doing, and not the university. And most people don't want to grant licenses to the university anyway. They prefer them to grant them to our commercial partners. Early clinician input is vital. Luckily, a lot of our projects are clinical academic led, so that comes with it. But if not, it's really good to try and understand whether the modalities are suitable for the disease, and whether actually a clinical study is feasible in the indication, and that's really important. And the other bit of advice is try and choose the right partners. Make sure they're aligned with your vision. Make sure they've got the expertise to turn your vision into a therapy, and also the significant resources that are required to do that. And the last thing is that kind of the wider our network of contacts, the better we get at supporting projects. And so if I've not spoken to anyone here before, I'll be very happy to talk to you later. Thank you.
Hello everyone, my name is Julie Warner. I'm Vice President of Regulatory Affairs at Boyd Consultants. We are a drug development consultancy specialising in the development of advanced therapies. So my background is in physiology and molecular immunology, and I've been in regulatory affairs for about 19 years. So we really work a lot of the time with small organisations, often virtual companies and university spin-outs, to help translate these great ideas into the clinic and help them progress along, get funding, and hopefully reach the market. So one of the things I want to talk about today is my journey here, I suppose, in terms of navigating a roadmap. So I actually am really grateful to be back in Bristol. I lived here for a year, many years ago, and spent a lot of my time driving around the streets of Bristol with an A to Z clamped between my teeth. By the time I left after a year, the top right-hand corner had just about disintegrated. So it's really interesting for me to be here talking about a different kind of roadmap. So when we think about navigating the path to the clinic, I think there are quite a few analogies with one of my hobbies, which is running. So stay with me. I think it's planning and good preparation are key to success. Because when I think about starting out on a long race, and I usually get cajoled into signing up, I always think about the end goal. So a lot of people will be familiar with the old mantra, fitness is a journey, not a destination. I actually want to present the opposite for regulatory affairs and helping to progress projects through to the clinic. It's all about the destination. It's where you know you need to get to or you want to get to. So be that conducting a first-in-human study or moving a product through from first-in-human to phase 2 or phase 2 onto phase 3. If we know where we want to get to, we can plan out the journey. We know what steps we have to take, and then we can take some actions to de-risk that journey. And I'm lucky enough to work in early development now, so I'm going to walk you through an analogy with racing. So for me, when I'm planning a run, it's all about the right fuel at the right time. So this is what a typical race looks like for me. That red dotted line is what I know happens if I don't plan accordingly. If I don't take on fuel at the right points, it's going to be messy. It's not going to be very pretty. I'm not going to enjoy it. I'm going to feel tired very quickly, and I probably have quite a high chance of not actually finishing the race. When we translate this into the development setting, that's really what we want to avoid. We want to avoid crashing out very early on when we could have taken steps to prevent it. So if we compare this to drug development and we think about starting at the end where we want to be, we then work back along the pathway and we make sure that we inject the fuel at the right times so that we stay off the critical path. And by fuel, that's regulatory intelligence, be it from advisors who are experienced in the field or by going and talking to regulatory agencies. And this process does take time and it takes a lot of planning. But if you start at the far end of the graph and think, for instance, that might be a first-in-human study, that's where you want to get to. In order to get to that point, you need to know some aspects of the trial that you want to run. Then if you will have those key aspects locked down, you know the kind of non-clinical studies that you need to run to support that study. When you know the kind of non-clinical studies that you need to run, you know how much material you need to manufacture, you know when you need to manufacture it, and you know what quality it needs to be manufactured to. And then there are various steps we can take throughout that process to make sure that we keep the risk out of the equation. So thinking about the development of advanced therapies, we see quite a few hurdles, for want of another athletics term, and we see a lot of challenges and hurdles for projects that specifically come out of academia. And believe it or not, the biggest hurdle is actually manufacturing. Everyone always thinks it's the clinical trial or some of the non-clinical studies, but actually when you take everything else out of the equation, it's the manufacturing. Because if you don't have a product, you can't do your non-clinical studies and you can't run your clinical studies. So that's why we always try and work backwards rather than forwards, because then we have a realistic timeline and we have a realistic plan. So two critical issues that always come to mind when I'm thinking about manufacturing and looking at projects that come out of university settings. And there's some great research that goes on, but we just need to make sure it's of the right quality that we can carry forward. So the first issue that always comes up is the quality and the provenance of the material. Sometimes we see projects where non-clinical proof of concept studies have already been conducted. Sometimes it's just an idea, sometimes it's a bit of a dabble with an experiment. But what's important is looking at where is perhaps that viral vector coming from? Has it come from a -80 freezer down the corridor in Tom's lab? Or does it have good provenance? Do we actually know what it is? And that ties into avoiding potential pitfalls later on. What we don't want to do is get to phase 1 studies or phase 2 studies and find out the material that we've put into humans needs to change because it's been fully sequenced. If we're talking about a first in human study, it's perhaps been fully sequenced and we found out that there's something in there that we didn't want to be in there because there was some contamination back in the lab and something got in there that shouldn't be in there. And then we have to either change the product or do a further analysis to justify the safety, essentially, to backfill the gaps. We want to avoid changes later on because they can be extremely costly, not only in terms of money, but in terms of time. And that's the critical thing here when we're talking about a lot of patients with a really high unmet need. We want to make sure that product gets to those patients as quickly as possible, and it is a good quality product. So we will always we'll always ask questions, where did your vector come from? Are there any intellectual property issues around it? And working with people like Andrew's group, we can avoid a lot of those issues up front, but it's one thing to always bear in mind. The second aspect relating to the manufacturing then is layering on what are the regulators' expectations going to be when they see an application for a clinical trial for this product. They will want to know how it's manufactured. They'll want to know what the quality of the product is. So how is it tested? What are the risks associated with the manufacturing process? Are the tests used to test the final product that goes into patients appropriate? Are they suitable? And do we know that they're fit for purpose? So can they repeatedly test different batches of the same product within a reasonable set of parameters? They'll also want to know how many batches we've made and what are the data on those batches so that they can know we have a fairly robust manufacturing process. And then they'll want to know how stable the product is. So can we keep it in the fridge? Can we keep it in the freezer? For how long? Specifically for advanced therapies, once it's, how do we defrost it? What happens if we have to extend the shelf life? How do we keep it when it's been defrosted? How do we safely make sure it can get from the pharmacy essentially into the patient? And we want to make that journey as smooth as possible when it's on site. The other things we also need to think about for manufacturing is the manufacturing process that's planned scalable? So does it support being increased in size so that we can supply further larger clinical trials and ultimately the market? And the manufacturing of some of these products is incredibly complex. I think when we started out working with simple gene therapies, I say that now, I can, but when we started out working on early ATMPs, obviously manufacturing was a big hurdle then, but a lot of manufacturing for some gene therapies especially is fairly straightforward. But now we're seeing a lot more complex advanced therapies come through, multi-component products. And at some point, you have to find a manufacturing facility that can mix all those components together and do it in a way that is good enough quality so that it can be given to a patient safely. So assuming we can overcome all of those hurdles, we then have to think about the non-clinical side of things. Now, as we all know, the development of advanced therapies from a non-clinical perspective is quite unique. There's no boilerplate approach that we can take, unlike a small molecule or even a more standard biologic. Quite often we are limited by species constraints, by the ability to actually deliver the product in the same way that we want to deliver it in the clinic. So we have to think, what's the best way we can safely evaluate this product in a reasonable way? What makes sense and what is feasible? And we have to just ground ourselves in the science there. And if we can put forward a logical argument to regulators for what we want to do, and they agree with us, that kind of rubber stamps our non-clinical plan, which makes things less risky for us. And then obviously there are challenges from the clinical side as well. If it's difficult for us to run robust, non-clinical studies that answer every potential question that we might need to answer before we go into the clinic, what risks remain for a clinical study and how do we mitigate those risks? Do we need to think about, for instance, any prophylactic treatment for patients so that we potentially avoid adverse events? How do we determine the starting dose that's safe? How do we know what dose we can escalate to? These are all kinds of questions that often need to be dealt with in an absence of information. We just have to apply good scientific logic. And then obviously when we think about a lot of advanced therapies, many of them have been in the rare disease space as well. So the difficulty comes in finding the number of patients that you need for some of these studies as well. And how do you draw conclusions on a very small data set? So many challenges, but I would urge everybody to think about manufacturing from the outset, because that is the longest timeline going. In terms of the right interaction and with the right regulators at the right time, this is critical for success of a program. When we think about approaching regulators to de-risk the strategy, what we really need to do is think about what we want to get out of that meeting. What purpose is it going to serve? Because if we can't answer that question, there is no point going to talk to regulators, because they aren't there to develop a product for us. They can comment on what we want to do, but we have to give them a reasonable plan for what we want to do. So there's a framework there for them to agree to. So we always think about that first. Then coming back to the source of the fuel, essentially, we think about which agency it's best to talk to. In the US, we have the US FDA, nice and easy, cradle to grave approach. Great, we stay with the same team all the way through. In Europe, it's a bit more complicated. We have centralized regulation with all 27 member states now. Don't make me sad. 27 member states now, but then we also have national regulation as well. So we can, depending on what you want to get out of the meeting, you might be better off going for national advice versus centralized advice, but that's something that has to factor into the equation as well. Then we need to think about what we want to ask the agencies, and we've probably got a good idea about that, because if we've gone through that exercise on the previous slide of planning out the development program, we roughly know what kind of questions we've still got unanswered, or questions where we think we're kind of deviating from what's been done before, or we're trying to push something forward in a really novel area. And we might need to do a bit of education with the regulators as well, but we focus on the questions that we want to ask. Based on that, we can prepare a good package, and as part of that package, we still have to look at what gaps there might be in the program. Quite often we find there are still gaps. There are some questions that we just can't answer, and we have to think, why is that okay? Why is what I want to do, this study, in the absence of knowledge on X, Y, and Z, why is that study okay? We then start to think about what we can do with the output as well. Because if we go through this whole exercise and we have a good meeting, we should be able to run our non-clinical studies, we should be able to run a good clinical study, and we should be able to get some data out which we can add further value with via further regulatory activities such as orphan drug designation, early access measures, and that kind of thing. So really the success of a program is pinned by interaction with the right regulators at the right time. Of course, nothing ever goes to plan. So there will be bumps in the road. Don't be afraid to recalibrate. If you have to go off plan, you can readjust your regulatory roadmap at any time. But if it's not there, you can't adjust it. But hopefully there should be success at the end if we follow these basic principles.
Good afternoon, everyone. I'm Carolyn Porter. I'm CEO of CytoSeek, but also here in my capacity as one of the organising committee of Southwest Advanced Therapy Networks. So in the immortal words of Monty Python, I'm going to talk about something slightly different. And what we were trying to do in terms of covering different topics on this panel was to give an indication of what were the ingredients for success for translation in the region and how we access them. So I'm going to talk about financing opportunities. As you might expect, I'm going to give an example of financing opportunities for early stage businesses by looking at Cytosec, the company that I'm CEO of. So I'm going to start with an introduction. Oops, there goes the Southwest Advanced Therapy Network Committee. For those of you who aren't aware who's on the committee, come and speak to us afterwards. I'm going to start with an introduction to Cytosec, for those of you who aren't familiar with the business. We are a spin-out from Bristol University, and we were established to commercialize the synthetic biology technology platform called artificial membrane binding proteins, or AMBPs. These are designer proteins that can rapidly add enhanced functionalities to cell therapies, and we're going after one of the big challenges for cell therapies, which is the treatment of solid tumors. We're testing our AMBPs currently in combination with 2 cell types, CAR T and NKs, but our technology is cell agnostic, applicable to autologous and allogeneic cells, and also anything with a lipid membrane. So we're getting some interest from LNP synthesizers and also from cell therapy manufacturers as well. In terms of our financing journey, we didn't quite reach the lofty heights of Pure Spring with a 45 million single investor, but we closed cumulatively just under 5 million in a mixture of seed and non-dilutive capital. And that's what I'm going to dig into, just to give you an illustration of the types of capital that are available in the region to those who are starting their journey. So starting off with the beautiful Cytosec color, Cytosec pink and blue. On the top is sort of a history of our seed financing, and which happened over kind of 3 stages, if you like. But just to mention, before 2017, which is when the narrative starts, at least on this slide, as with many other spin-outs, it was the accumulation of 10 years research in our founder Adam Perman's lab, and this is very typical for most spin-outs. So the first funding that Cytosec took was what I've described as hypothesis testing. This is from UK I2S, which is a small government-backed fund that's managed by London-based Midburn, and that came in the form of a convertible just to test the hypothesis. Could our AMBPs get on cells and actually enhance the functionality of those cells? Oh, I can see some of the words we could be doing. Apologies for that. And following that phase, our founder, I think, was deliberating whether he wanted to spin out, and Andrew can share more of that story as he was along to the right, and preparing really for a potential spin, filing IP, filing publications or Nature publications, and also engaged with an entrepreneur in residence in the Cambridge ecosystem to really advise on where could the technology add value. It had a provenance in Regen Med, but really the biggest opportunity was seen in the field of immuno-oncology, where solid tumors were the nut yet to be cracked. So through the course of 2019, the team, some of the founders are here in the audience, were successful in securing an Innovate non-dilutive award that required equity matching. So that crystallized our first kind of pre-seed round with a combination of Innovate funding and equity that was raised from UK I2S as well, who's been an enduring supporter of the business in our young years. Also Parkwalk, which is one of the biggest UK investors in spin-outs, and also local angels. So I joined the company in 2020, and a little bit about my background. I've worked for 20 years in different roles in big pharma and business development, and also in corporate finance and early stage fundraising. And one of the things that attracted me to the region was what's happening in the tech space. A bit of sense of it from Ashley early on, was resources and capabilities to support young companies, and also a lot happening in the cell and gene therapy space, which is why I've put myself forward to get involved in trying to create these networks. So my role was really to come in and close the seed round. And 6 weeks after I joined, we had lockdown. So our fundraising was done through the medium of a Zoom window. But for us, having local investors as part of that narrative was really important, because they had familiarity with the founders, the technology and the business, and they cornerstoned our round. And we went on to close an oversubscribed round. We went out to raise 3, closed at 3.6, and we had 4.5 on the table. We brought in some new investors, notably Science Creates Ventures, who you heard about earlier on from Ashley as well, an important constituent of the Science Creates ecosystem, and we were their first investment and also the first tenants in this facility. We also brought on board non-regional funds, including Meltwind, which is Jonathan Milner's fund, who's building a portfolio of cell and gene therapy companies. Interesting on the left, just a couple of comments on that. This is not the allocation of shareholding of different sources of funding, but really the percentage contribution the different sources of funding have made to our raise. And significantly, over 40% are based on EIS funds. We still have a significant angel component on our cap table, and 10% roughly non-dilutive and 10% venture. We expect this to significantly change as we approach our next raise. But as mentioned at the bottom of the slide, 50% of the funds raised were from funds that were based in the region or had a regional association. So this was really important for us through lockdown, where we weren't able to get out and see people in 2D. So Cytoseek isn't the only organization or entity in the region that has raised capital in the last 12 months. It's just an illustration of those, and apologies to those outside the Bristol ecosystem, a number of them are Bristol based. And I think what's interesting is the investment in infrastructure capability and manufacturing capacity. And I'm illustrating two other examples there of funds that were raised in 2021 in NHSBT. They were part of a successful 18 million bid for 3 sites to build a gene therapy hub. And the funder there was like a LifeArc and MRC. And are those in the audience who can speak more eloquently about the capabilities that are built in that arena. I encourage you to speak to them afterwards. And also Exmoor, an important constituent of our ecosystem, and also a member of the Southwest Advanced Therapy Network Committee. They closed 12 million raise from private equity funders, MBM, to expand their GMP and process development capabilities. And I believe from Remy, the structure is up and running, and hopefully will be in place by 2023. I think you said in your press release. I'll hold you to account. Also wanted to mention, in addition to funds that have come in and stayed in the region, that we're as a region contributing to UK Gene and Cell Therapy PLC, for want of a better word, with IP being licensed from Cardiff into Anara, which is about TCRT therapies. Pure Spring we heard about earlier on, sadly the 45 billion has made it out of the region, but hopefully some will come back in again in due course. And Mobrify, who Andrew mentioned earlier on, who we share investors with. So illustrating kind of the breadth and depth of capital, both on the non-dilutive and dilutive side. So this table just summarizes that to illustrate, if you're thinking of starting a business, there is various diverse sources of capital that are accessible to you and to enable you to grow. Some are very much regionally focused, like Bristol Private Equity Club, and we have some investors from that entity, and sometimes Creates Ventures, who are located in this building, but others are very much international or outside the rest of the UK. So just one final point before I sort of finish up. Fundraising, and particularly in a capital-intensive environment in the cell and gene therapy arena, is not just regional. Regional was important for us as a small business through lockdown because we had visibility in the regional ecosystem and they cornerstoned our land. But there has been a lot of activity and investment in the space, and Andrew already signposted this. And this is just an example of beyond seed and some 10 million plus raises that have been closed in the cell and gene therapy sector in the UK. And I think what's interesting is we're seeing an increasing interest from corporate VCs and also the blue chip US investors such as Arch and NEA. So in other words, if you're in this space, the future is bright, whether you're at a regional or a national level. Finally, I'm just going to conclude with a couple of points. It's been a good 12 to 18 months for our region. I think various diverse sources of funding have come into the ecosystem. As you will have heard from Andrew, there's more companies on the way. There is foundational IP flowing out of the region, but we're contributing to UK Cell and Gene Therapy PLC as a region. So there are funding levels that are accessible up, of course, a range of different types. And I think the intention for our region is only going to grow as more spin-outs come and hopefully as we progress our own fundraising cycle. And as I mentioned before, although regional fundraising sources were really important to us and are accessible to businesses starting, we're in a very capital-intensive ecosystem. And so national and global funding and sources are going to be important to us going forward. So hopefully that's given you a flavor of one of the sets of resources that are important for success in our field and are accessible. And I'm going to hand over to Axel to talk about arguably what's the most important part of the translation journey, which is getting cell therapies into the clinic and into patients. Thank you for your attention.
Right. Hello. Hello, I'm Max Walter. I lead the trials unit at the Bristol Cancer Institute, or Bristol Haematology and Oncology Centre, which is always a bit of a mouthful, but Bristol Cancer Institute doesn't include James and his haematology colleagues. So it's but it's the same building. It's a bit behind the Children's Hospital at the BRI. Do you remember 2017? Before lockdown, isn't it? So a few highlights. There was Trump, there was Grenfell, there was the royal engagement, and we even had our own fire at the BHOC. But, and that's really where the link comes in, it was the year that the first CAR T-cell therapy was approved by the FDA. So there have been lots of research going on, but the first time a CAR T-cell therapy was approved was in 2017. Now, looking at James, he's going to tell me that's probably not true. She's excellent. And the reason that matters is there was a building excitement, but actually there was nothing in the clinic yet. The one thing that didn't happen in 2017 was us recruiting to a CAR T-cell trial. We have been ready to go end of 2016. We were all set. We had the trial open, but we had backed the wrong horse. It was a Junos product that unfortunately, out of the 9 patients in the phase 1 in the US, 5 eventually died from brain complications. And it raises the first thing is you need to have a product that is safe. As a clinician, I cannot put something into patients that isn't safe. And it comes back to Julie's point of manufacture. If you produce something that isn't reliably, the same stuff, then that gets me slightly anxious, and the complications that happen is a different matter. But it needs to be safe. So we didn't have the CAR T-cell trial. However, we did have one of those many, many, many expressions of interest that we fill in as clinicians. Would you like to take part in this study? And I said, well, it sounds fun. And we set up a phone call, which is where the TEL comes from, the telephone call with a chap called Robert Brown from Iovance, who talked to me about a TIL study in cervical cancer. And I treat ovarian cancer predominantly, but I also do systemic therapies for other gynecological cancers. So cervical cancer falls into this category. And their trial proposition was about take a patient, you take a tumor piece out, you take that to the labs at the bottom of the slide, you separate the tumor-infiltrating lymphocytes, and from the cancer cells, you purify the tumor-infiltrating lymphocytes, you make more of those, around 100 billion to be precise, and then you put that back into patient under step 4. And before that happens, they also have had non-myeloablative lymph depletion. So you create a niche into which you can put those new lymphocytes. And then in step 5, you give them interleukin 2 to really give them a thing at the backside so that they can start to attack the cancer. And then hopefully the patient recovers and gets discharged. So I've told you about those first 2 steps. That was in May 17, I filled out this thing. And actually really quickly thereafter, I had this discussion with Rob Brown. And, there's a lot of groundwork that then follows from that, because much as we had set up and then didn't recruit, unfortunately, that CAR T study, TILs were different. So there were discussions with the CTU, our clinical trials unit, with hematology, and James was instrumental in supporting that study with us. But also the surgeons would have to get the tissue out, because that was a novel step. The ward, can you support the patients? Can you look after them? Do you have capacity? Research and innovation, who sort of run the research side on our stuff, and have to give permission for us to actually do the study in the first place. You then have things like a site selection visit, where they say, yes, we want to use you. You have a research forum where you say internally, do we have the capacity and the capabilities that we can deliver on what we want to promise? And then eventually you get to the site open. But actually that sort of site selection visit to site open is where the real work begins, because you then have to run around like a blue bottom fly and sort it all out. So you have the clinical trials unit, you have the contract research organization who runs the study for the sponsor, but you also have the sponsor, and that's the first hurdle, where as a clinician, typically our studies, you have a molecule, whatever compound it is, chemotherapy, we sort of know how it all works. We ask a question, and the CRO have done it lots before, they answer the question. If you have something as complex as TILs, that way you have to have chain of custody for the sample that goes from us to the sponsor, and then again for the product that comes back to us, at what point do you hand over? So there's every specific question. CRO may not be able to answer that, and suddenly it becomes Chinese whispers, because you have to ask the CRO, they answer the sponsor, they answer the CRO, they answer us, and you go, but that's not the answer to the question that we asked. So a good link between us and the sponsor was useful enough, and we agreed with that they would answer those specific questions directly rather than how we do it. But there's also lots and lots of other stakeholders involved. It's slower than planned, but there's the ATIM committee who needs to sign off on the research in the trust. There's a gynae team, or whichever clinical team you're in, you have to say, well, actually, we support this as a therapy for our patients. There's radiology who say that they can identify the lesion that you want to resect. There's a bone marrow transplant team who ran all this for us, because actually a lot of the processes of treatment is much more akin to autologous bone marrow transplant than is anything I had ever done in my career. And so with the support of hematology, we were, yes, they're our gynae patients, yes, we follow them up, yes, we have the input into their care whilst on the ward, but actually the day-to-day running of the myeloablative lymphodepletion and the infusion and the IL-2 decision-making about giving the treatment was made by the hematology team. There's a surgeon who has to take it out, the pathologist who have to do the frozen section on the day to say, well, actually, there's cancer in the sample. The hematology team who then look after the patient, there's the water team, and for all of this, you also have ITU on standby, because it turns out IL-2 is not entirely non-toxic. I think in the US, they had 40% admissions to ITU. They have capacity, they're perhaps slightly more keen to fill that capacity. For us, it was one patient, and I think probably 10% is a more reasonable figure with the IL-2 doses that we used. But you need them on standby, whether it's 10% or 40%, because at the end of the day, you don't want to suddenly have to argue with them in the middle of the night when the patient needs to come in. Let's say if it's a cancer patient, it's a trial patient, you don't know what to do. And just to give you an example of how these interactions all work, ITU took 3 months to get on board. Not because they weren't really keen, but you need to speak to the research lead, who then speaks to his team, who then say, yes, we're interested, please tell us more. So you go and give a presentation. By the time you found that meeting in everybody's diary, it takes time. Then they go back and talk about it again. And then it just takes time. It's not that they were obstructive in any way, shape, or form. It just takes time. And then we also had our first patient on the 13th of June. Got harvested on the 3rd of July, and then she was meant to be treated on the 7th of August, but because she had mycoplasma in her cells, we couldn't give the cells, and she then got reharvested and treated on the 22nd of October, which again is testament to everybody being really engaged, because we had robustly set up all those pathways, and had agreement for everyone to say, actually, no, we can turn this around quickly again. So recruit and treat, and the hard work continues, because of course, you have to run around again for a patient to make sure that the sample that comes out of the patient really reaches the sponsor in a way that you want to. You need to make sure that you have a process in place that when the sample comes back, it comes on dry ice. What do you do with the dry ice? You have 24 hours to keep them. We had planned for 24 hours, but you can keep them dry ice for longer. But once it's thawed, you really need to go in relatively quickly. So you need to then coordinate it. So I'll give you the trial journey quickly. So the consent on, this is not the first patient, because this is the patient where the journey worked more smoothly. So 30th of August, it worked. You didn't have 28 days for screening, cardiac function, lung function. For this patient, she needed a staging scan to assess where the disease was. Sometimes the prior CT scan is in date, as it's called, so no less than 4 weeks old, and you can use it. But a CT head was additional. Bloods, ECG needs to meet the surgeon, needs to have an anesthetic to read to make sure that it's actually safe to even get the sample out of the patient. That this has nothing to do yet with can you put it back into the patient. It's just can we get stuff out. You then have the harvest. You need to then re-establish suitability to make sure that the patient still has something to see on the scan that you can measure, because there's no point in putting something in if you can't measure the effectiveness. Then the BMT review comes in, so the bone marrow transplant team who were giving the treatment, as in the chemotherapy to lymphodeplete, as well as then the IL-2 after the infusion. And then you get authorization, only then do you start lymphodepleting. So you do not want to start with the protocol, but I guess generally, you do not want to start lymphodepleting if you then haven't got the patient to give anything back. And for this particular patient, she got her lymphodepletion, she got her TILs, she got all doses of IL-2, and she was discharged 6 days after starting the IL-2. So 3 days of IL-2, which made her reasonably unwell, and then actually recovered really quite quickly without significant complications. All of this only works if you have a very large team. And apologies to anyone who was involved and who is on this list. Jack at the back is, I know you're on the list, as is Helen, who is also somewhere in the room. But James is on there are lots and lots and lots of people, but actually, on top of the individuals named here, there's the wards, there's the ward doctors, there's the consultants, there's intensive care, there's trials in pharmacy trials in. So And of course, the patients, because we only treated 2 out of the 7 patients that we treated, only 2 were our patients. The rest were all referred in, and they agreed to travel. They agreed to come on that journey with us, and without their sort of enthusiasm, I mean, it's their life. You might say, well, it's their license. It's sort of, they want to be treated. But if you have a choice between, I haven't got that long to live, do I come to a team that you're going to do something that isn't proven, or do I spend that time with my family? It's still a decision that they need to make, and they place their trust in us. So thank you to all of them. So what have we learned? Cellular therapies are complex and resource intensive. There are the demands from the sponsor. So there's lots of data that you would want as a sponsor, but of course that means that the clinical teams also have to deliver a lot of that data. And it is more than it would be normally. Normally you treat on day 1, you get 3-4 weeks off, you treat again, you capture the toxic side effects that have happened over the 4 weeks, that's all. Here, patients are an inpatient, you daily blood tests, daily, all of that needs to be reported, and it's a lot. There's coordination of the teams on the day. You get the surgeons and someone to take the sample to pathology, who then say, yes, let's come down, and who then puts on dry ice, who then gets it to the courier. It doesn't just happen because you think it should happen. There's a number of stakeholders involved, and I showed you some of those. Novel therapies have novel ways of not going to plan. I don't want to say go wrong, but again, with chemo, and that comes back to Julie's manufacturing point, you expect the chemotherapy to be there. It's in the fridge somewhere, or even in a cupboard somewhere. With the cellular product, it needs to be made somewhere, it needs to be shipped in a sort of time-critical manner to arrive when you come. So for us, it was mycoplasma. For us, it was someone not getting the cells because they just didn't grow. I suspect there's other things which can go on. That's before you then consider any of the novel side effects that those therapies may have. The patients may deteriorate during workup. We had one patient that we treated, we probably, in retrospect, felt that we shouldn't have done, because things got slightly late and she kept sliding. It's a difficult conversation to have. Consented, harvested, but we really shouldn't be treating. We didn't have that. We got her through it. She survived, but in retrospect, it wasn't. But that's a learning curve as to how do you manage that, because that's not something that we normally have to do. Once a patient is on the study, within 3 weeks you start treating. Here it's within 10 weeks, 12 weeks sometimes, and that's different. Robust pathways, risk assessments, and SOPs, standard operating procedures, minimize risk. You need to be a stickler for detail. You need to train lots of people that then know what to do, because in the middle of the night, when it's an on-call senior house officer, like one junior, and a registrar from home have to make decisions. And yes, of course, they get consultant backup, but the consultants aren't in the hospital overnight. And I certainly can't do 24/7 when I have a patient. It just doesn't work. So everybody needs to be trained. You need to have a quality agreement with the sponsor. In other words, who's responsible for what? You take the sample out of the patients, you put it on dry ice. At what point is it no longer our responsibility? At what point does it become the sponsor's responsibility? How do you track that? You have, there's a secure ID, there's SEC ID. How do you get one of those? They're an HTA standard, but how do you get one? You may need temperature monitoring, always do, but for the ION study, it was much tighter than it was on our regular fridge. So we needed to think about how to get a freezer that actually matches their requirements. You need to build expertise for future studies, because having done it once and having double tagged pretty much everything you could do just so that we made sure that we didn't get it wrong on the day, you cannot run a trial that way. You need to build the expertise, and that comes back to any one of you who wants to spin out and looks for sites to run a study. You want to have sites involved who know what they're doing. And we were lucky to get the study because they were looking for someone in the Southwest. We could say we're doing CAR T cells at a time when there wasn't that much expertise around, and that's why we got into it. And we now got ION Therapeutics, we got a basket study from them, we've got a new lung study, we're doing more CAR T cell studies, we're doing the inducible peripheral stem cell studies, we have the dendritic cells. We have expanded quite a lot to build on all of this, but that doesn't happen overnight, and you certainly don't want to take it. In this day and age, you need to be careful where you take a punt on a site. You don't always want to go to the same sites, but you probably don't want to run a trial with 5 sites, none of whom have done this ever before. And we now have an advanced therapies trials manager, Helen, at the back, who was actually involved in the setup and has taken a lot of that expertise over into the where we are now. Right. Someone's taking out my slide. I was going to show you one slide as to how well it's worked. Anyway, there was a lovely waterfall, but out of the 27 patients in the study, 12 had what's called a formal response, so met the criteria about how much the cancer had to shrink before it's called a response. But out of the 27 patients, only 6 patients had cancer lesions that were slightly bigger after treatment than they were before. So they are exciting treatments. Actually, stuff can work, but they're non-trivial, and it will require more work to get them into the clinic. Thank you.
Great. So thank you very much to the speakers. I think there were some excellent bits there, and I'm hoping that we can step now into a bit of a Q&A. So what I was going to suggest is, if the speakers don't mind coming up, we'll have 2 chairs on either side. So hopefully the people that are online can also see. And I'm hoping that there may also be questions that are coming online. So if you are at home listening to us, hopefully you have been able to both see and hear us, which it proves sometimes to be remarkably difficult. But please do put things online, and we'll head up from there. If anyone in the room wants to ask a question, you can just raise your hand. As chair, I was going to take the prerogative of asking 2 things that I'm going to go through for everyone. The first one was, where is the main pain point in the area that you discussed? Where, if you could have a magic wand to change something, what would it be? Julie, in terms of that regulatory pathway, if you could change something, what would you change?
I don't know if I'd change anything about the regulatory pathway, but I think I would urge everyone to make sure that expectations are realistic. We see some quite unrealistic timelines of expectations, I think, for that time from manufacturing to I think it always takes longer and costs more. It's like building a house. It always takes longer and costs more than you think it's going to be. Make sure it's well processed.
The pain point being that expectation. Carolyn, in terms of funding, what's the biggest pain point? What would you want to change?
Well, I think despite painting a rosy picture about access to capital in the region, the depth of capital, we're just for our Series A at the moment, and part of the reason I'm passionate about highlighting this region is to convince large-scale investors that we have the capability to deliver outside of the Golden Triangle, where a lot of the expertise resides. And so it's great to hear that you guys have run some of the first clinical trials in this ecosystem. So I think being visible and access to significant capital. I think as a small company in this region, also access to talent. We've been really lucky being able to attract talent into this ecosystem, and part of that is being part of this infrastructure and broader region. But I think going forward, there is a skills gap in the cell and gene therapy region more broadly. So as we execute on our next raise, access to that talent is going to be a challenge for us.
So, and I think that there's a lot of additional things going on here. Obviously, in the last round of Advanced Health Science Centres, Bristol was the only additional centre that was added in, and I think that sort of is a real testament to what we can do in the region. Obviously, we go further afield as well, and I think particularly drawing across from Bristol to other areas, I think is going to be important. Axel, in terms of the trial and setup and all of that hard work, what one thing would you like to have been able to change?
More capacity, so we can take more on more of the studies.
So I think there's quite a bit at the moment. There was a letter only a month or 2 ago from Lucy Chappell as the scientific advisor for NIHR, lead for NIHR, talking about reset after COVID, and clearly a lot of NHS organisations at the moment are struggling to deliver those clinical trials that we absolutely need, both for patients and for our the companies and universities in the region to be able to get products forward. But hopefully as things go on, that will improve. But yeah, absolutely, I'd agree, capacity remains a problem, and I think that's all stages of this, whether it's the trials unit, whether it's the clinical site, whether it's services for collection, for laboratory services, they're all constrained at the moment. And Andrew, in terms of your work, what one thing would you magically change?
I can't say the draft funding system, but if people have ideas, then sometimes that's quite often a grant. It could be a year before this starts work, and that doesn't lend itself to rapid development. Have development use, so I would like to see more development and then more translation. Easier for us to invest more technologies.
speeding up that very first step, really, of the process. I know Carolyn has a question. Does anyone else have a question in the room that they'd like to ask, or we can keep it to the panel at the moment? I would encourage people to ask questions if you'd like. But Carolyn, would you like to ask a question?
Yeah, I mean, my question was for James and Axel about the likelihood of first-line therapies, cell therapies being first-line therapies going forward. I mean, the complex process that you just described with TILs and the sort of headcount resource and complexity of managing that would suggest that that is still somewhat in the distance. And I know there are some indications where we're seeing cell therapies in particular as second-line treatment regimes, but I just was wondering on your observations on how far away you think that is.
If I start on that, Axel, are you happy? So I think there is a lot to be said about starting in the relapsed or refractory setting, because usually you can get your results much more rapidly. But we also know, particularly in the CAR space, that once you've had more chemo, collecting adequate cells and healthy cells can be more difficult. And also, probably the responses are less good. So I'm UK Chief Investigator for a CAR-T trial using CAR-Ts for myeloma in the first-line therapy. So it's CARTITUDE-5, and it's patients that we have determined aren't going to have standard of care therapy, which would include an autologous transplant of their own cells. And instead, they're going to be randomized to receive CAR-T cells or not. And the really interesting thing for that trial is we're looking at people in their 70s, often 60s and 70s, to go into that trial. So we're looking at cell therapy at that stage, and hopefully for the companies, it'll work out, because actually getting products in at frontline is important. I think right from the beginning, when this was being discussed I was at meetings in Parliament about it, and one of the questions that came up from those were, well, when are we going to start using these early? Can we collect cells at least at the beginning and store them and use them for generation of cellular products later? And at least the Novartis CAR-T cell product, Kymriah, you can actually store cells, I think it's 3 years in advance. So actually there is a potential to collect cells when people's cells are healthier and younger and utilize them later. And one of the trials of CAR-T cells run by Celgene actually does that. They collect cells from patients immediately after their frontline therapy. And if they relapse, then they've already got the cells there. And I think Axel really showed that one of the problems of cell therapies has been this length of time to get people onto treatment. And we've had people with standard of care therapy who progressed to the point we couldn't treat them. And I particularly remember a woman who was in her early 40s who got sent to us despite being in a really poor clinical state, and lost a day that she could have had with the kids because the referring centre weren't happy to tell her that she couldn't have the therapy they needed to send her to us. So I think anything that we can do to reduce that length of time, whether it's some of the technologies that will come along with allogeneic products, off-the-shelf products, or whether it is over bringing therapy into the front line or pre-storing cells. So I think, yes, absolutely, things are already moving in the direction of earlier stages. And I'm sure Axel will say it's similar for cancers that he looks after, but we've seen products come in at 4th, 5th line, move forward in the treatment paradigm that we're following. And so now for myeloma, which is the main cancer that I treat, we've got daratumumab in the front line. A few years ago, that was 4th line.
Yeah, I think you spoke most about hematology. So for the solid cancers, there's a distinction between curatively resected, because surgery remains a huge part of what we do for solid cancers, and the adjuvant setting. I think that is relatively long time away at the moment. Although people with the checkpoint inhibitors have really pushed to try and get them into the adjuvant. But if you have a reasonably high chance of being cured with standard therapy, to prove that you're increasing on that cure rate is a trial that takes 5 to 7 years. So that is going to be a delay. I think in the metastatic setting, that people like James said, it seems to be that the fitter you are, the more likely immune therapy is to work, and therefore why not use it earlier? But there are other challenges with solid organ malignancies. We're just not as far with the products yet. But if you have something that is better than chemo, which clearly doesn't cure people once you have metastatic disease, why not try immunotherapy?
Any hands? Terry.
What's the success rate or conversion rate, or the number of projects that you start talking to those that actually make it all the way through to successful?
Just in case that hasn't been heard online, it may have been, but I'll just repeat it. So it's looking at how many of the initial discussions with Andrew end up actually making it through to a spin out or licensing.
They're probably, I think, across the sector, it's about five disclosures to any one deal. I think that in the ATMP space, because of the market for it and the strong interest in the field, it's more like one-to-one. But that's obviously that's all right things. But there is demand that. I mean, the problem is that once we've done the deal, we're not then in control of what happens. And so I'm really hoping that those deals do actually lead to some benefit. But
Susie, were there any questions online?
So I wanted to read out one of the first questions that came through Julie. When do you think a phase 1 study should move to phase 2, and what aspects? That's a good question. The development of progression from phase 1 to phase 2 depends on several factors. And the package, the base package we generate in phase 1 will probably need to be supplemented using a non-clinical perspective. Phase 2, we want to be the readouts. You might want to study within a different country, so you might have to put regulators in those jurisdictions to make sure that there's a number of things. The critical thing with our therapies is sometimes we're looking at very long follow-ups for patients, because there are so many unknowns at the moment about what long-term consequences might be of therapy follow-ups for patients. And we know some information, but we don't know all of it. And it's still really makes us the critical thing is making sure that those patients follow it for a long time, but we don't have to wait 10 years to reach there is a
I think that's experience that we've seen in lots of cases, and I think, particularly mentioning from Axel's talk about the fact that we were supposed to be opening a trial called Orbit from Juno, you can see how you can end up with a problem. It looked so promising in preclinical, it looked promising in the initial trials, and then there was a change in the way that they were making the cells, and then they had this massive neurological toxicity and multiple deaths. And actually, at the same time, so many other companies were also entering the space that they decided to just bin that product completely because they couldn't turn it around and went on to their next product. So, and I think from a hospital point of view, one of the things we really learned from that was that actually it takes a lot of time and effort to set these trials up. And in general, the income comes when you recruit your first patient. And actually now I think we're much more savvy to that and asking for setup costings, because we can't afford within the hospital to have a load of people's time that is effectively lost if the product then doesn't go forward.
Excellent. My name is Madhu, and I'm mainly targeted. Try to develop a lot of combination. I was wondering, adding low doses
Possibly, but any cytotoxic therapy that you give will ultimately also affect the CAR T cells and is mildly suppressive. So there are concepts of what can you prime cancer by inducing mutations and then therefore by increasing it, so making the tumor immunologically hot. Theoretically, but CAR T cells are so specific to one single epitope that probably CAR T cells are not the thing to go, but it would be my take.
No, I would agree with that. So thinking about combinations, and we look even at the just standard transplant field, and adding in immune modulation afterwards can be potentially a route forward. So thinking of things like lenalidomide as an immune modulatory agent alongside allogeneic transplant has been studied, hasn't yet been reported, which makes me worry that it has quite a bit of toxicity rather than benefit. But actually that combination of non-toxic therapies is becoming more common, whether that involves a gene therapy or a cell therapy. There aren't many yet with that. And I think we've obviously talked, or I've talked quite a lot about CAR T cells because they're already there in the clinic. You've got to remember that even internationally, there's probably only about 10,000 or just slightly less patients that have ever had a CAR T cell for any indication. You compare that to a trial before you get regulatory approval for a tablet used in heart disease, where you may need 30,000 patients in the trial. We are still right at the beginning. And that's what makes it so exciting. That's what makes it so interesting. We do not yet know the best way of delivering these therapies. You know, the trials at the moment, we've got a trial of the CAR Ts used in adult ALL from Autolus. And in that trial, they're splitting dosing if there's a lot of disease, in part to try and see if that reduces toxicity, but it also may change the kinetics of memory formation and longevity of the treatment. You look at the initial products that are on the market, they all use the same sensing domain within the CAR, but actually you might be better off with a different one. You may not want to bind quite as well. They went for a really high binding capacity with that, but actually you may end up pushing all of those cells too far to be effector cells and not getting as much of the promise that we had for CAR T cells, that they're a living drug, they're going to stay around for years and provide immune surveillance. But if you actually make them attach a bit less well, you do get that memory potentially, at least in some of the trials. That's obviously looking at the cell side. When it comes to gene therapies, and there's so many different things to talk about in terms of how they're delivered, and the same within those CAR T cells of actually how you get the genes into the cells and what you do with that. So, I think we're really early, but I think probably you will find increasingly trials that then look at what you do to the patient before they get a cell therapy, and what you give potentially alongside it, and then really also what you do afterwards, because we're still in a position where if a patient relapses, what should we do? Should we do a transplant? Should we consider CAR Ts again? Do you have a new trial with a CAR T with bispecificity? Can you do all those different things? Do we have a question in the room? Yeah. Does anyone have anything they know and can share with the room? Or
I don't know whether it's been raised in
when I did my PhD, one of the people who was doing a PhD in the same lab was looking at inducing FOXP3 as a regulator of a suppressor T cell, with that exact thought that actually you could utilize those for autoimmune disease.
I think it's imminent.
Are we doing something for the myelin multiple sclerosis patients in there?
Well, so there is a trial. It doesn't use an advanced therapy. It's just obviously the use of autologous transplant has become more popular for autoimmune disease. So there's STAR-MS trial in the UK looking at that for multiple sclerosis. There's also a trial looking at it for inflammatory bowel disease. So clearly, cell therapies, whether they're advanced or are standards, have an ability to reset the immune system. And so absolutely, you can see the science behind it would suggest that it is a route that we may go for. And when it comes to commercialization, obviously some of those diseases are big and have a big pot of money to go after.
Yeah, so these are expensive. As I understand it, that the artist over things over £200,000 of treatment. So, and there is this perception that you know, the NHS, these are not that easily affordable for the NHS. So therefore, the UK not an attractive. That's a perception. I just wondered, you know, does the cost of these therapies come up in fundraising? And I wonder also if it increases the pool of sponsors for the trials.
I think there's quite a few parts to that. Yeah, you are completely correct. The list prices for the CAR Ts that are out there are sort of $290,000 to $300,000 just for the cells. The associated costs with actually providing that in the hospital are upwards of £90,000 per patient on average. So I don't know what deal NICE managed to make with the companies, because we don't get told that, but NICE seems to be a government-sanctioned shakedown agency rather than anything else. That's what they're there to do, is to get the price down. So yes, they are expensive, but you've got to think about the comparisons on it. So if you have, you know, some gene therapy products, for example, where the costs of treating a patient with that condition over the course of their life could end up being £10 million, a £2 million gene therapy that's curative when they're a child saves the NHS 8 million. The thing that's a bit more difficult is in the cancer area, where we're currently giving these cell therapy products, where it is actually potentially a life-saving product rather than a saving of a lot of other costs. Now, with the CAR T cells coming up front in myeloma, the hope is that then saves money on the vast expenses in the field for chemotherapy going through multiple lines. At the moment, it's not clear that would be curative, though. So it may just delay that expenditure, but then it's what price do you put on a human life? And governments around the world struggle with this. We have cost per quality output for NICE, and actually the agreement is that the cost per quality that's allowable is greater for cancer therapies than for other therapies. So when it comes to cost, you know, the NHS, the most cost-effective thing, I think, when they've done the cost per quality, turns out to be aspirin. So we could sort of stop everything else and just give people aspirin, and then we're fine. But it's part of that whole discussion about what the benefit of saving someone's life, keeping somebody alive for longer is.
But you are changing. Sponsors raise it as an issue.
Not particularly.
Because by the time they come to us, you know, they've got the money in the bank and they just, you know, hand over the cash to the trust.
Well, when we're commercializing technologies, sometimes what we do is get health economic analysis undertaken. So licensing in the market there, but typically they don't understand that and do that because that's what they do. But it is an issue.
It's still a field that's investable. So we're targeting CAR-Ts and speaking to investors that this place for investments in that arena. And I think if you look at the two kind of lead CAR-T programs, they made record returns for their angel investors and their foundational investors, the exits that they created. So those are kind of good news stories for the sector, but there's still money going into our programs. So, and we do get routinely asked, what is our technology to cost of cell therapy? So it is a consideration for investors. It's a consideration for us that we're working on it.
I know.
As we prepare for our next raise.
And I think it's really interesting though when you say, you know, people get £300,000 for the cells. Most pharmaceutical products that come through have a profit margin of like 95, 98%. The profit margin on a CAR T cell is only about 50%. Now, some of that relates to the massive investment that's had to go in terms of infrastructure. And so for the companies that have that infrastructure now, it won't be as expensive to get their next product through. But it may lead to changes in actually how you look at the whole field. And some of the newer cell therapy companies coming on are looking much more at going away from that centralized model of production and potentially looking at some of the benchtop machines that are coming out from Miltenny with the Prodigy or Terumo got a new platform as well, which the name escapes me at the moment. But actually, that may well shift the dynamics of how expensive these products are. The Americans are also talking about hospital cars, so looking at development of things locally. Obviously, then you still need to make sure that you don't infringe on IP. And the question is, how much proof do you need of equivalence of a product? These are so complex that if you don't infringe on the IP, do you change the properties so significantly? And for a hospital to do a trial of a CAR T that they're making themselves probably also becomes prohibitively expensive. Yeah, absolutely.
So it's a general question about whether sponsors are being definitely not in the UK. We've seen a lot still very invested in the UK and still very, like I said, the UK government has done an awful lot to foster research and development and really work with sponsors to get them far quickly and make sure that they're helping speed it up.
And I think one of the things that I might just mention, because otherwise my director who sat at the back may complain, but there was for hematological cancer and transplantation, a trials network set up about 4 or 5 years ago called Impact. Impact was funded by Anthony Nolan, Leukemia Research and NHS Blood and Transplant to help deliver these cell therapy type trials. We didn't really get many cell therapy type trials during the initial period, which was sort of a proof of concept. But we have a network where there is a funded research nurse in each hospital site. And we have now invested in the new vehicle that's going forward, which is called ACT. And that's actually a non-for-profit private enterprise with CRO functionality built into it now. So if people do have cell therapy trials, particularly in the blood cancer space, that's somewhere where there is expertise and help to get your product into lots of patients quickly within that network. And the investment that's made from it will also be able to be reinvested back into investigator-led trials in the UK infrastructure. So definitely something, if anyone wants to ask further about it over drinks or by email, that would be fine. I had 2 very eager people at the back.
So I have a question about you touched on that, and that was a long-term follow-up. So like one company, especially springs to mind when I think about long-term follow-up, that's Uber Bio, because they had quite a constant product, and they're looking at these bars, there's like an awful lot of requests put on them for regulators, pilots, following up and which to manage it to end up with the product. Do you think, does that happen a lot? Does that put a lot into Spinouts off? That there might be like the complexity in the fact that a lot of the people we're dealing with are ill, so they're not going to manage 20 years follow up, and the actual cost that we have to kind of put into this for manage these follow-ups and packaging as well. Does that put a lot of these companies on?
Stage, I don't think it does. I think we've got quite a good system now. Kind of go back to what we were saying earlier about detaching the long-term follow-up from an initial investigation. So, you know, there are ways and means to pull patients over into kind of extension protocols and things like that. So the cost of running those extension studies is a lot less because you don't have such intensive monitoring. It's done at more infrequent intervals, etc. You know, different periods of time over which you're expected to follow up. Well, so for some gene therapies, patients only follow for 4 or 5 years. Lentiviral vector will be longer. So I think it's very much a case-by-case basis. But I don't think it does put a lot of companies off because there is some ways and means that we can still meet the requirements of quality. I think your point about ultimately is this going to be a viable model at the end of the day? I think with some of these, especially small patient you know, it comes back to returns, and is there enough there for the company to actually make this viable commercial that they can still get some funds coming back in to reinvest as well. So yeah, I think it's only because we can only make a best effort, but I think the role naturally is.
Can I just add to that? I mean, the reason they the drug companies are much better at setting prices than the governments are keeping them down. I mean, I don't want to take away from what you said. I think, and certainly last time I looked, which is a few years ago now, pharmaceuticals were the only growth sector where the growth margins have been more than 10% year on year, in the capitalization of the companies. They're still better at making money than we are running them in. But run a trial, clinical trial phase 3 costs 50 million or so. And if then at the end of it, it says your drug doesn't work, you go, that's 50 million gone down the drain. So, but at that point, you need big money, you need to have a plan as to how do you run all of this, at which point the follow-up, you find ways of making that cheaper. But that if you've got that far, that no longer should be a deal breaker. When you are at the site to seek stage, someone saying, "Oh, we're not going to put 50 million into this," because you have absolutely no idea. I hope it works, but you have no idea where this is going to go. By the time you're in a phase 3, you have at least a phase 2 study with 150 patients saying, "You know what, that might actually come off."
I know. I think one of the advantages that we've seen from these therapies, the cellular therapies being given in effectively a transplant team setting, is that the transplant teams are already used to the fact that they have to follow up people for the rest of their lives. And so therefore, transplant services have data managers, they have quality managers, they have everything already there on the clinical side. The more difficult thing is actually what data do you need to collect for that time? And at least with the commercial CAR-T products, their post-marketing or authorization required them to follow up the patients given CAR-Ts. There still hasn't been agreement on exactly what data is going to be collected and for how long. And the company signed contracts with EBMT, the European Bone Marrow Transplant Society. They still haven't told us what they're doing. So we've ended up with a lot of ad hoc data collection, which actually does then put a lot of pressure on to teams. One thing that is also different is that way of monitoring formally counts as research, and therefore we're having to probably go back and reconsent all the patients in a different way than they were consented initially to ensure that we got that sort of rollover, even though it's going to be part of standard of care, but that rollover sort of research consent from patients. Sorry, the question back was first, then I'll come to you.
And my PhD matrix real issues with I was just thinking about
So I'd say we're not really doing that in the hospital. That's being done in a cell manufacturing facility. And in general, those have become quite centralized. There are programs in London where they have been producing them through the university, sort of attached to the hospital. And obviously there can be difficulties in that. I transduced cells for 3 years, and for human cells, I was very happy if I got sort of 4 or 5% initial transduction, and then you've then got to expand them, and then you get problems like mycoplasma and other things, and that wasn't in a GMP type level. So it's something that we have within NHS Blood and Transplant. We've got 4 GMP rooms up in Filton that have a grade A hood and a B background, so we can produce these things. And we've also got very high quality production of things that can be used for research, the peptide vectors. We're talking about obviously the lentivirus and adenoviral bits. We're working on those through our Biotechnology Centre, which is just moving from being next to a field of cows in Langford up to Filton as well. So I think there is a real understanding that these are difficult products, and getting them right from the beginning, as Judy mentioned, is so important. But yeah, that's why you then have to define your release criteria at the other end of it in terms of what you can have. And we've had with CAR T cells quite stringent release criteria that were required by the EMA. But then when you've got a patient waiting for it, it's amazing how many of them get them released anyway on a concessionary basis afterwards, because actually often the numbers that were picked were so strict that actually it makes no difference whether, you know, we had one patient where overall the number of cells was completely appropriate, but the viability was just below what had been defined in the release criteria. And so, you know, from a clinical point of view, there was no reason not to give those cells. But obviously from a regulation point of view, we then had to jump through several hoops. The good thing for the NHS is if they're outside of spec, they're free. Sorry, you had a question.
Yeah, hi, Naj. I'm software developer. We're in advanced therapies. My question is around data, Axel, but I think it relates to everything that the team has spoken about. It's cost of goods, it's the shortage of QPs worldwide right now. Also the manufacturing, consent mechanisms to kind of handle patients long term, whether it's 10, 20, 30 year follow-up. Decentralizing those consent mechanisms so the patients are really patient-centric. These technologies are around today. I think as a sector, we seem to be talking a lot around the fact that we are digitally, we're going to have a digital transformation at some point, but surely we should be digitally native right now. From day dot, we should be doing this from the design, from the outset, from drug discovery, all the way through to commercialization, market authorization, etc. We're struggling in that space. And I would say that as a developer, a software developer, I want to be connected. I mean, I did this for far too many years, like I said, like large pharma. I know too well how sensitive cost of as well for the industry. But really, to bring the cost down, a lot of those costs are embedded in the trial space, cost of goods in logistics, manufacturing, how do you operationalize and streamline scaling up or scaling out, whether it's autonomous or allo. It's the data piece, and it was implicit in the chain of identity custody condition point to make it. How do we do that? How do we make it better? That is a big gap, and that will affect everything.
I think your point about we should be digitally native, I agree entirely, but the NHS, don't forget, is a large socialist government organization. We are not digitally native, I can promise you that.
That's what we're more successful in Germany.
Yeah, they throw more money at the problem, but they're just as fragmented as a market. So, but the other thing is that it's not only that the NHS isn't quite there, it's also a lot of the biotechs that come, you know, that we do trials with, because they all have a different electronic patient record that they want data entry, and they have a different this system and this system, and they don't all talk to each other. And then, you know, we suddenly have to be fully integrated with 10 different systems to cater for all our trial sponsors. And then we may do one trial a year. If we know we're going to do 50 trials with someone over the next 10 years, yeah, fine, we might even do that, but not for one trial when it's difficult enough as it is. So I think there's an element of, yes, we as a health service provider aren't set up, but the community out there also can't agree on their standards. No, you just make do with different systems. That we are used to, and what I described about the double tagging of every step of the way, that is not sustainable. I mean, I'm with you on that one. But as you get better, the person who collects the sample knows what they're doing. And okay, at some point, you know, they won't even need the hand holding of putting it into the ice bucket. The surgeon can do that. You know, it's not difficult. You just need to know what to do.
And data is a big thing. There's a report that will be coming out at the end of next month, which is the UK Stem Cell Forum report into the future of transplantation and immune effector cell therapies in the UK. And data is one of the important parts of that. With myself and Dan are both work group leads for that report. And we have had a data workshop, including NHS Transformation, which is the new NHS Digital. And actually, they're looking at quite a few bits of it, which I think fit in quite nicely. Data is so key to everything we do, and we don't collect it well in the NHS. It's also worth a lot of money to people. And actually, if we collect it well, it makes us a better environment for then going out to big companies around the world and saying, come to the UK, come to the Southwest, come to wherever to have your trials, to deliver your product. And so I'm quite certain that things will be improving. The other interesting thing is that actually this region is part of an NHS transformation pilot into something called a trusted research environment, drawing things together, which I hadn't heard of until I happened to be on a call where someone mentioned it. And actually we're looking to explore whether we can take sort of transplant cell therapy, some of the work we do at NHSBT, into that setting. We are actually out of time, or rather my bad, sharing has taken us slightly over, but some really interesting questions, some fantastic talks, I think everyone would agree. For people online, you can go and make your own cup of tea, and I'm sorry that we can't share further conversation with you, but if everyone could give their thanks to the speakers today, and if you have further questions, we'll be outside in just a minute to take them.
Thank you very much.